Greetings, everyone. Once again, I want to welcome you to my YouTube class. In today's class, we're going to be talking about another nematode parasite that affects man and other animals. And the name of this parasite is tri- Trichinella or Trichinella. That is the genus name, and the species name have many species. We have spiralis that affects man, then we have Nesoni nativa and pseudospiralis that affects other animals. So that's the parasite we want to study today. Now, when it comes to the classification, the phylum is Nematoda, the class is Adenophoria, the order is Strachyrata, the family is Strachinellidae, then we have the genus as Strachina. And like I said, the spiralis, the Nelsoni, the nativa, the pseudospiralis are other species of this parasite. Just a minute. Okay, now when it comes to the characteristics of the these parasites, we have the fact that they appear like a thread. These are some of the characteristics. They appear like a thread. They are thread-like in appearance. And apart from that, we're told that the parasite lacks leaves and buccal cavity, unlike what you see in some other nematode parasites. Then we also have presence of stickosome. The stickosome is a kind of a complex of the glandular cells and the pharynx. They collectively form a structure that is called stickosome, and it's peculiar. Is as a matter of fact, it's peculiar to Trichinella, and it's one of the diagnostic features of this parasite. Then each cell has a single gonad. You know, it's either the testes or the ovary. It's just one. Then there's presence of one or no spicules. For some species, they have spicules, and that spicle is a structure that helps in the transfer of sperm in males during copulation. Then, okay, the size of the size of the female is usually twice the size of the male, showing that the females are always larger than the males. So that is the characteristics of this parasite. Then when it comes to the life cycle, the first thing I want to understand the life cycle of this parasite is that the parasite is homozygous. Homozygous. And when we say that the parasite is homozygous, that means that, that parasite requires one host to complete its life cycle. The opposite of homozygous is heterozygous, meaning that the parasite requires more than one host to complete its life cycle. So, for the case of Trichinella, sorry, it requires one host and it is homozygous. This host could be human or animals. So the life cycle of this parasite begins by the ingestion of raw or undercooked meat containing the larva of this parasite. The larva is usually situated within the muscle cells, that is the nerve cells that kind of provide proper conditions, good conditions for proper development and growth of the parasite. So any nest cell that is carrying any muscle cell, sorry, carrying Trichinella is usually referred to as a nest cell, a cell that takes care of the parasite, just like nurses take care of patients in the hospital. So the life cycle of this parasite begins by the ingestion of raw or undercooked meat. So when this raw undercooked meat is ingested, then excitation, because it's usually in the encysted form within the muscle cells. So when it is ingested now, it excists. That means the cyst covering the parasite breaks, and once it breaks, the larva comes out of the small intestine. When the larva is released within the small intestine, then it molts severally. Remember the pattern of development in nematode parasite from the infected stage, which is L2, it molts to become L3, from L3 to L4. So when it gets to L4, which is the fourth stage, the fourth larval stage, then what the parasite does is to invade the mucosa and the submucosa. Within this region of the small intestine, that means where it has invaded the mucosa, the submucosa, and that is where they mature again, or kind of develop further into adult. By the time the adult males and females are formed, then they mate, and after mating, they produce larvae. Now, it is known, or it has been studied and discovered that each female produces about 1,500 larvae. And once the larvae are produced, the males die. As a matter of fact, after mating, the males die. And the period between the injection of this larva and mating, and also production of the new larva, is usually within 30 to 32 hours. This is just we can just call it 2 days, approximately 2 days, within, the period between the injection of the larva and the mating, and also production of new larva from the mother parasites. Now, once these larvae are produced, then the larva will now undergo what we call the liver heart lung migration. It begins to migrate from the liver to the heart, from the heart to the lungs, and reaching other sites within the host body. But finally, it settles within the muscle cells, and that is where it remains until when next an individual, you know, uh, takes such animal where this parasite is hiding or sees itself, then the cycle continues. So this is the life cycle of Trichinella spiralis. This parasite causes a disease that is called trichinosis or trichinellosis. And when it comes to the pathogenesis or pathology of this parasite, it can be studied in different headings. Now, the first heading is what happens when the fourth stage of the larva, or the adult one, penetrates the mucosa or the submucosa of the intestine of the host, and the part of the intestine is the small intestine. Of course, when it happens, you agree with me that it's going to cause lesion within this region where it has penetrated. And apart from lesion, when I say lesion, I mean wound. Apart from that, it could also result to secondary bacterial infection, and such area could be inflamed, resulting to inflammation and nausea and sweating and other things that may be associated with it. Then, apart from that, first this first part I've mentioned, which is the effect of the adult one, we also have the effect of the migrating larva. Like I said, this larva undergoes migration, the liver, heart, lung migration, and it migrates to other parts of the body. And as it is migrating to such sites, it also causes inflammation. Like for instance, it gets to the lungs, and it inflames the lungs, resulting to pneumonia. Can also cause accumulation of fluid within this region where it passes through, resulting to what we call edema. It can also inflame the liver, resulting to hepatitis, that is inflammation of the liver. That is the second stage. Now, the third stage is the effect of encysment of the larva. Remember, I said that this larva, exists within the muscle cells, and that is because the muscle cells are the cells that enable the proper growth, and provides a kind of nutrition and nourishment to this parasite. So this parasite grows, this parasite, sorry, is nourished within the muscle cells. So within this muscle cell, that is where a physiological process is triggered within the parasite, which results to the encysement of the parasite within the muscle cells. And the invasion of these parasites within the muscle cells also triggers a kind of immune response by the host, and this immune response usually involves the infiltrating leukocytes, such as the macrophages and the eosinophils. They are usually triggered when this parasite invades the muscle cells. And after that, what the parasite now does is to insist within the muscle cells. Now, this encysment can also occur in other areas where this parasite has gotten to, maybe in the kidney, resulting to nephritis, in the brain, resulting to meningoencephalitis. Sometimes it could also insist for those that pass through these other parts of the body and they are stayed there. It can inflame such areas or such organs or structures in the body, and the inflammation of the heart can result to what is called myocarditis. So this is the effect, or these are the effects of the encysment of the larva. That is for pathology. Then diagnosis. How can this parasite be diagnosed? One simple way is, or one common way is through muscle biopsy. Muscle biopsy, bio and C. Biopsy. That is I kind of assaying or investigating the muscles. Muscles are collected and pressed between glasses and examined under low power magnification. With such process, one can see the larva of Trichinella spiralis. Then we also have other ancient method of diagnosis such as complement fixation, precipitin, and flocculation, which are not majorly used now. They are no longer in vogue. But other methods such as serodiagnostic methods such as the use of ELISA, that is enzyme-linked immunosorbent assay, is also being used for the purpose of diagnosis. However, the most effective method of diagnosis is through the muscle biopsy. Then treatment. How can this parasite be treated? This parasite can be treated using a mebendazole. That's the most effective drug when it comes to treatment of trichinosis or trichinellosis. The disease is either called trichinosis or trichinellosis. Now, apart from the use of mebendazole, corticosteroids, which is a kind of drug that helps to reduce pain, it can also be administered because of the inflammation that this parasite has caused in the body, in the intestine. It could cause a kind of pain. So, in addition to mebendazole, the class of drug called corticosteroid can also be used, and they also have purgatives that can also help to expel the worm. There's a class of drug that's called a purgative. One can also take it so that it will help to expel the worm. So this is the way that this parasite can be treated. Then for prevention and control, it's always good to make sure that every meat you take is properly cooked. If it's properly cooked, then of course the heat will destroy the larva of the parasite before getting into the body. So, and we'll have other methods such as humans making sure that he or she does not interject himself within the sylvatic chain. Of course, we'll have two different types of trichinosis. We have the sylvatic trichinosis because this parasite also affects wild animals. So humans should not interject themselves within this sylvatic chain. That's another way of prevention and control. Then, of course, we also have urban trichinosis. That is the one that affects, domestic animals. So if you are to take domestic animals, you should also be sure that domestic animal does not contain this parasite in its muzzle. And of course, just cook properly. If the meat is properly cooked, it will go a long way to kill the parasite and reduce the transmission of this parasite. Ladies and gentlemen, this is where we'll stop the class for today. In my next class, we're going to talk about other nematoparasites. Like I said, we'll have other nematoparasites that we have not touched, as Caris rubicoides. We'll have Encelosoma dodonecata americanus, Strongyloides saccharialis, Wucherea bacroti, Loa Loa, and so on and so forth. So as time permits, we're going to be talking about these parasites one after the other. See you in my next class.